to Mr. Wanyoike, the digital learning platform. In front of you is Mr. Wanyoike himself, ready to present to you a topic in genetics. And in today's lesson, we are going to look on the first law of heredity, that is, which was defined by the Mendel. And therefore, before I come to the first law of heredity, I am going to define several terms, and one of the terms that I'm going to look at, various terms, and the first terms which I'm going to look at is the first one here. We have RL, the second one is homozygous, the third one is heterozygous, the fourth one I'm going to define dominant gene, the fifth one I'm going to talk about a recessive gene, and also I will explain something to do with genotype. The seventh one is the phenotype, and finally I'm going to look on monohybrid inheritance. And I'm going to start with the first one. And that is RL. RL refers to a pair of genes. This is a pair of genes that which are supposed to be located on the chromosome, and therefore a pair of genes is what we refer to as an RL. And the second one is homozygous. As far as homozygous is concerned, this is a condition where the allelic genes are identical. And therefore, whenever the allelic genes are identical, that condition is referred to as homozygous. And an example of homozygous condition is the gene that represent the height. For example, tallness, the characteristic of tallness. It can be represented by two identical genes or two identical genes like that. This is capital T or small t, and later on I'm going to explain what do we mean by the capital T's and the small t's. I'm going to explain that. And therefore homozygous, as I have said, it is a condition whereby the allelic gene are identical. What about the heterozygous? On the other hand, heterozygous is the condition in which the allelic genes are not identical. And therefore, if the allelic genes are not identical, in that case, that condition is referred to as heterozygous. And an example, I can come up with capital T and small t as far as the characteristic of tallness is concerned, and therefore I can represent it that way. And therefore I'm going to go to the fourth one, and that is a dominant gene. And therefore what is a dominant gene? A dominant gene is that type of gene that expresses its characteristic fully, both in homozygous and heterozygous condition. And therefore, whenever you are asked what is a dominant gene, you are supposed to say that a dominant gene is the type of gene that expresses its characteristic fully in both homozygous and heterozygous condition. And the second one, for example, if I come to the whole dominant gene, you can see that capital T here, if it was an individual having these two types of genes, these two identical genes, it will appear tall. Physically, it will appear tall. And what about this? Physically, the individual which have this kind of a combination of gene in its RL, that is too small t, it will be dwarf. It means that small t represent dwarfism. And on the other hand, the dominant gene, capital T, when it is grouped with another one, it occurs together with small t. This is what we refer to as heterozygous. I have already explained this. And capital T is a dominant gene, and small t is a recessive gene. And therefore, what is a recessive gene? A recessive gene, it is the type of gene that only expresses its characteristic fully only in homozygous condition. And therefore, as far as a recessive gene is concerned, it cannot express its characteristic in in heterozygous condition. It only expresses its characteristic fully only in homozygous condition. And therefore, in heterozygous condition, a recessive gene characteristic is masked with the dominant gene, and therefore the dominant gene in presence of dominant gene, a recessive gene cannot express its characteristic, and therefore we normally say it is masked by the dominant gene. And therefore a recessive gene, once again, it refers to the type of gene which will only express its characteristic fully only in homozygous condition case. For example, that characteristic of height. And therefore, if two recessive genes occur together, if the allelic gene is represented by two small t, and therefore that individual will become dwarf, and therefore the dwarfism it is observed physically, and therefore they will be having a short status. Their physical appearance, they are going to be dwarf or short in status. And what about a genotype? Genotype, as you can see here, the word genotype refer to the genetic makeup of an individual. So the genetic makeup of an individual is what we refer to as the genotype. And what about the phenotype? Phenotype, on the other hand, refers to the outward appearance of an individual, or the physical appearance of an individual. And therefore, in an exam, whenever you are asked to define the term phenotype, you are supposed to state this, that it is the outward appearance of an individual. And as far as the eighth term is concerned, that is monohybrid inheritance. And as far as monohybrid inheritance is concerned, it is defined as the inheritance of one trait or one trait which is controlled by a pair of hereditary factor. In other words, it is controlled by a pair of genes, and the pair of genes are both contributed by both the parents. And therefore, once again, if you are asked what is a monohybrid inheritance, you are supposed to say that this is the type of inheritance which involves the transmission of just one pair of contrasting characteristic. For example, we can give an example of monohybrid inheritance, the characteristic of height. We can have height individuals, and the contrast of a tall individual is dwarf individual, and therefore the characteristic in monohybrid inheritance, it must be one single characteristic, and also it is contrasting, and therefore the opposite of tallness is dwarfness. And therefore, as far as height is concerned, that is a monohybrid inheritance. Another example, we can give the shape of the or the texture of the seed. You can have smooth seed coat as far as the seeds are concerned, or wrinkled seeds. Also, that is a monohybrid inheritance, and therefore it can be classified into that monohybrid inheritance as far as texture is concerned, as far as height is concerned, and therefore these are the examples of monohybrid inheritance. And I'm going to come to the next area of study, and I'm going to introduce another area, and that is during genetics. We normally study genetics using various methods. We can use methods of determining the genotype of an individual. So we can have various methods, and the various methods that we normally employ so that we can study the genotype of an individual. The method number one, we can use genetic cross. Genetic cross. And the second method, we can use Punnett. Punnett square method. And the third one is we can use, that is genetic cross. We have Punnett square method, and the third one we can use the pedigree diagram. Pedigree. So there is a D there. Pedigree diagram. Pedigree diagram, and therefore these are the three methods that are normally used whenever we are studying genetics. And therefore, whenever you are asked, you identify the methods that are used during the study of genetics. You say the first one is genetic cross, and the second one Punnett square, and the third one is referred to as pedigree diagram. Having said that, I'm going to look on Mendel's work as far as the father of inheritance is concerned. He did some work which enabled him to be called the father of inheritance. And some of the work that he did, work done or experiment done by experiments done by Mendel. The first experiment, he made a genetic cross between two dwarf garden pea plants, and therefore he crossed two pure breed of garden pea plants. And therefore, as I say, pure garden pea of dwarf varieties, of dwarf variety. And when he made his work, he observed that after his work, and he repeated for several generations, and the conclusion was that all the offspring obtained in each and every generation were all dwarf. And therefore, in his work, he was crossing I can use a genetic cross to demonstrate genetic cross to illustrate his work. And in his work, he made a genetic cross between two dwarf garden pea plants. And as far as these two crosses are concerned, the results yielded that from the results in all the generation were TT. The genetic makeup of all the offsprings were small t's, and therefore meaning that all of them were dwarf. And therefore, when I can use the genetic cross, to explain this, what normally happens, you are supposed to separate the parental genotype. They separate, and that separation is what we refer to as segregation. And once they separate, there is that one and the other one here. They separate that way, and also the same case applies to the other one. That is, there is segregation or separation, and after that, this is the parental. We can talk of parental genotype, and they separate, After separation, these are gametes. The encircled ones are gametes, represent gametes. And the gametes, they are supposed to be represented singly in a single gamete. And therefore, you can see each and every circle is having one small letter T. And there is random fusion, whereby this may fuse with this one, that is T. And this is random. There is random fusion, and this is the F1 generation. I can say generation. F1 generation is represented by the results there. Then we have this one may also have been crossed with, because it is a random fusion, meaning the first one goes that way, the second one may come, may be linked also with the second one, and therefore we'll have the second offspring. And the third offspring, this one links with the other one, and that is T small. And also the second one may link with the second one on the other side, and therefore you'll get a T. And therefore, what is the observation? You can see that the genotype of F1 generation, all of them are dwarf characteristics. The genotype is represented by all of them have a genotype of represented by two small t's, and meaning that all of them were dwarf. That is the genotype. That is the genotype of F1 generation. The genotype of F1 generation is represented by small t, two small t's. And therefore, as far as his work is concerned, you can see that the result for F1 generation, even for F2 generation, even after he repeated the same experiment for many generations, he obtained the same result. And therefore, he concluded that whenever there is a dwarf characteristic, that a dwarf characteristic is supposed to be homozygous codation. It is a homozygous recessive codation, and therefore you can see that the parents normally transmit their genes to their offspring, and therefore all of them were short. And what about the second Mendel's work? In the second Mendel's work, the second experiment, in the second experiment, Mendel's worked with a tall variety now. And the tall variety was not pure, was an impure tall variety, meaning that I can delete this and explain what happened. And therefore, as a result of Mendel working with the tall variety now, you normally know that tall variety, we have, it can be represented either in homozygous codation or heterozygous codation. And therefore, there is that, and also there is this one. That is homozygous and heterozygous. Homozygous 2 capital T, and the second one is having a capital T and a small t. And therefore, the first one is a homozygous dominant, and the second one is a heterozygous dominant. And therefore, if you make a cross between two homozygous dominant codation, a cross between two of them, you will eventually find that all of them will be tall. And therefore, in his study, he observed that he obtained a mixture of the two. And as a result of obtaining a mixture of tall garden pea plant and dwarf garden pea plant, it means that in his experiment, he used an impure tall variety, which was in heterozygous codation, and that is not a hybrid. It is not a hybrid, and therefore, that's why whenever you make these a cross, you cross with another tall, and the law of segregation applies here. We have a capital T, and we have a small t there. Also, this separates. This separates, and once they separate, you will get a T capital there and a T small. These are gametes. And this is segregation. This is the segregation, and this is parental genotype. We can talk of parental genotype. And as far as after segregation is concerned, the gamete fuses, random fuses, and therefore that can go with that. That is at all, and this is random fusion. This is random fusion, and this one may link with this one, and this becomes like that, and this may link with this one, it becomes that way, and this also may link with this one, and this becomes small t to small t, and therefore you can see that there is a mixture whereby the phenotype of the resulting F1 generation, you will find that these are tall. These are tall plants, and finally we have another feature here, and this is a short. This a short or dwarf. You can talk of dwarf garden pea. And therefore, in the second experiment, he obtained tall variety versus short or dwarf varieties. And as a result of that, he concluded that it was not pure. The garden pea was not pure. And therefore, he selected now from the tall variety. He selected some seeds and also grew them together and also made a cross between the tall variety and tall variety. And he repeated this experiment for some time until he was in a position of obtaining pure variety. And when he as a result of he obtained the pure variety as a result of crossing, that is crossing, that is experiment number 3. In experiment number 2, I have said that he obtained a mixture of tall variety and dwarf variety. What about in experiment number 3? He selected the tall variety now, the tall variety now, and he made a cross between them, actually pure. The pure ones, and therefore he made that, and for example he made that a pure breed and another pure breed, and after cruising he was in a position of obtaining that. He realized that all of them were purely tall. It means that after he made that cross, he obtained that all the of F1 generation, the F1 generation which was obtained was of tall variety. Observe that he obtained only tall, and therefore, whenever you make a cross between a tall variety and a dwarf variety, for example, if in his experiment, I said that in the second experiment he used heterozygous, 2 heterozygous crosses. That is, one was heterozygous condition for the tallness, and the second one was also heterozygous. And in that experiment, he used a tall plant. That is a tall plant that is in pure state. That is pure hybrid. That is for the gene that determine the tallness. That is 2 capital T. And he crossed it with a dwarf variety. That is a small t. And after that cross, he obtained in that experiment, he obtained all garden pea obtained were tall. Let us see from this cross whether they are going to be tall. And therefore, I've said that the gametes random fuses, and this is capital T, and also this may fuse with this one because it is a random fusion, and this will become that way, and this may fuse with this one, becomes that way, and also this may fuse with the second one, and that will become that. And therefore, this is a random fusion, and this is gametes. This is segregation, separation of genes, segregation. And here, as far as number 3 is concerned, there is a parental genotype, and this is parental phenotype. We can talk of parental phenotype here, phenotype being tall, and tall garden pea plant, and here dwarf. And after that cross, it was observed that all of them were tall, and therefore the F1 generation, this generation was all of them were tall. And therefore, as a result of all of them being tall, that is the F1 generation. F1 generation, as far as F1 generation is concerned, he observed that all the offsprings were tall phenotypically, and therefore this he concluded that whenever there is a homozygous cordation case being crossed with homozygous dominant case being crossed with a homozygous recessive case, he observed that the results were all tall, meaning that in the results in the F1 generation, the dominant gene which is contained in the offspring dominated over the recessive gene, and that's why all of them were tall. And therefore, in experiment number 4, in his study, experiment number 4, he crossed tall offspring planted with the resulting seed, and therefore after he did the third experiment, as far as the fourth experiment is concerned, he took the F1 generation, that is the seeds from the F1 generation and planted them, and also he cross-pollinated them with the pollen grain of the same plants. He did what we refer to as self pollination, and therefore after self-pollination he noted that that is in test number 4, he took what we refer to as selfing. He did selfing, and as far as selfing is concerned, he took the F1 generation, that one of it, and self-pollinated them with the same pollen grain, with its pollen grain that is the same. And he realized that after that, you will realize that, let me come up with the genetic cross first, then we make his conclusion according to him, and you will find that is what we are going to get. And therefore after getting this, we'll get the F2 now, F2 generation, and this also becomes T. This is supposed to be a small t, the first one. This is capital T, small t. This becomes a capital T, small t, and this becomes a small t. And therefore, you can see that from his work, as far as selfing is concerned, he was able to obtain the number of tall garden tea plants, garden pea plant which were tall. The ratio was 3:1 as far as the dwarf garden pea plant is concerned. You can see that this one is represented by this one is crossing with this. Sorry, there is a capital T here. It should be a capital T because the capital T is randomly fusing with the capital T, and therefore you can see that the first offspring was supposed to be tall, and if the second offspring resulted, also it was tall because of presence of the dominant dominant gene capital T, and therefore the third the same, and therefore the 3 offspring are tall, and therefore phenotypically they are tall. What about the last one? The last one is dwarf, and therefore as far as the fourth experiment is concerned, it shows that whenever heterozygous cordation or individual with heterozygous cordation is crossed with another individual with heterozygous cordation, as far as the gene for tallness or any other gene is concerned, the resulting offspring must have the ratio of the dominant gene will be expressed, the dominant gene to recessive gene will be expressed in the ratio of 3:1, and therefore 3 individual will have inherit the the dominant gene characteristic, while one of the individual inherit the characteristic of dwarfism or the characteristic of shortness. And having said that, I'm going to come to the next area of study, and I'm going to state, according to this work, the Mendel was able to formulate the first law of heredity. He was in a position of coming up with the first law of heredity. You can see he did this work. That is, he did the first experiment. That is crossed two pure breed of garden pea plant. Then the second one experiment, he crossed. He made a cross between two impure garden pea plant, and the third one he made a cross between a pure garden pea plant in tall with homozygous dominant case condition, and another one with dwarf. And the fourth experiment he made selfing. And therefore, after this, all this work, it is very evident. It is very evident that he formulated a hypothesis or a law. He was able to formulate hypotheses and laws. And the first law that he came up with is the first law of heredity, and it states that the characteristic of organism are determined by internal factors or hereditary factors which occur in pairs, and only one of the pair of such factor can be represented in a single gamete. I repeat again, it states that the first law of inheritance states that the characteristic of organism are determined by internal factors or hereditary factors which occur in pairs, and only one of the pair of such a factor can be represented in a single gamete. I'm going to give you one example here, a further example, and the example which I'm going to highlight is this one. I had already written it, and in this example I would like us to apply the methods actually, and the law of heredity, so that we can come up with the solution to it. And as far as this question is concerned, you are told that there is a heterozygous garden pea plant in heterozygous condition was crossed with a dwarf garden pea, and garden pea plant used genetic cross to determine the phenotypic ratio, and therefore a heterozygous condition for tallness, that is a parental, you come up with parental phenotype, and therefore this becomes capital T, small t, and then it is crossed with a dwarf that way. Then after that, you are supposed to you are supposed to say tall pea plant, and this is dwarf pea plant. Then after that, you write the parental genotype, and parental genotype is this one. Then you close it with a dwarf like that. Then you apply the first law of inheritance, that is the law of segregation. They separate, that is segregation, and after segregation we'll have the gametes like that being formed. These are gametes, and the same case applies to the other one. This is small t, and now we come to random fusion. You'll get like that. This is random fusion. Then we have F1 generation, and this one may have fused with the second one. This is t, small t, and this one may have fused with this. This is T, small t, and also this may have fused with that, and this becomes that way. And therefore, having come up with that genetic cross, now we can address the question now. We are in a position of addressing the question. How do we address the question now? As far as this question is concerned, you are supposed to come up with phenotypic ratio, the phenotypic ratio. And as far as the phenotypic ratio is concerned, how do you do it? Phenotypic, that is the outward appearance. And as far as outward appearance is concerned, that is the first part, phenotypic ratio. So you will talk of tall, garden, P, plant, is to what? Is to supposed to be equal to garden P plant, that is tall, is to dwarf. Dwarf garden P plant. And therefore, you just count how many talls. As far as the gene for tallness is concerned, you can see that the first one is tall, the second one is tall, the third one is dwarf, the fourth one is dwarf. And therefore, the ratio becomes 2 is to 2, and therefore you can simplify the ratio to becomes 1 is to 1. And therefore, as far as the computation is concerned, you are supposed to get 1 tall garden pea plant. You can write it as we had written, 1 tall. If you simplify, you will get 1 tall garden pea plant is to 1 dwarf garden P plant. And as far as that question is concerned, as far as the phenotype is concerned, you're supposed to get that answer. What about the second one? So the second one is the genotypic ratio. And as far as genotypic ratio is concerned, you just indicate capital T small t is to 2 small t's. 2 small t, and the ratio becomes also 1 is to 1, because you will get 2 of them are having the genotype of capital T small t, and 2 of them are having a small t, 2 small t's. And therefore, after simplification, you will end up getting the answer of genotypic ratio of F1 generation, being 1 is to 1, The third question that you're supposed to address, the first part of the question that you're supposed to address is that if the total population of F1 generation was 1,200 plants, determine the total number, number of tall plants in F1 generation. So the population you are supposed to say the number is equals to the probability. You get the probability of tallness supposed to be a half, because half of the plants are tall and half of the plants in F1 generation are dwarf, and therefore it is supposed to be a half. You multiply by 1200, and therefore you are supposed to get 600 tall garden pea plant. 600 tall garden pea plants. That is the answer. That is tall garden pea plants. And therefore, as far as that question is concerned, you are supposed to have calculated it that way. And therefore, the question you are supposed to link whatever you have covered in the law of heredity, as far as the law of heredity is concerned, and also you use a genetic cross so that you can be in a position of coming up with the with the results, that is the F1 generation. And F1 generation refers to the first children from the first parents, and therefore that is what we refer to as F1 generation. And therefore phenotypic ratio, as I have said, it refers to the outward appearance. How will the F1 generation plants appear? And therefore, from this genetic cross, you can see that half of them, because capital T and small t, it is in heterozygous case, and T capital is a dominant gene, and therefore it will suppress the recessive gene. And therefore, that combination of gene will help enable them to appear tall. And therefore, two plants appear tall, and also two plants appear dwarf. And therefore, the chances of having a tall garden pea plant is a half, and also the chances of getting, or the probability of getting a dwarf garden pea plant is also a half. Having said that, I would like to give you the number, the cell phone number, which you can always reach at me, and you can use this number, that is the cell phone number. You can use 254, that is the Kenyan code, followed by 724 901 639, and therefore that is my cell phone number. And therefore, for more information, you can always reach me using this number, that is positive 254 followed by 724 901 639. And also you can search me at YouTube using Mr. Wanyoike. You can search me on YouTube using the name Mr. Wanyoike. And till next lesson, bye bye.